In this video, we will discuss about the results of the strategy that we have discussed in the previous video, that whether this sTRA and CA19-9 express independently on the pancreatic tissue or they are expressed at the same position. So let us quickly revise that strategy. The strategy is that first they will stain the cells with the hoist dye that can stain the nucleus of the cells and one of the anti proteins, and thereafter this TRM monoclonal antibodies, and then scan the fluorescence and they get rid of this dyes, and thereafter stain the cells with the hoist and different proteins, and at the same time for this CA19 monoclonal antibodies. And after that they will again rinse it and then treat it with sialidase, and again uh, stain the cells with hoist and different proteins and TRM monoclonal antibodies. In the third case, they will get information about the monoclonal antibodies that are sialidase, that has the sialic acids on it, as they have treated with the sialidase and then react with it. So by comparing this third TRA monoclonal antibodies and first monoclonal antibodies results, they can be able to get information about the TRAs that has sialic acids on it. And in the yesterday I told that this antiprotein 3 could be this against the proteins that express TRA, but this can be any antiproteins. The idea of using different antibodies in different strategy is to ensure that the protocol works properly, or we can say that this act as a control to show that this steps works, particular steps works properly. So what they find out is that they did the six different micro tumor tissue micro array assays, and four of them were the patient's tissue, and two of them were xenograft developed tissues, patient derived xenograft. And what they have find out, they did this tumor micro tissue micro array, and the example of this is shown in the panel B, where they have analyzed a portion of this core 6, and they have checked, they have shown the representations where they have checked this PDX1 in the first step and this TRA, and in the second step they check this MUC5AC, which is almost MUC5AC, which is almost not visible, and this CA19-9, and in third step they check this B catenin and this sTRA. So, and after that they are able to find out the informations whether there is overlap or not. So when they use the CA19-9, but the results of the CA19-9 but not sTRA is shown here, this should overlap with this fluorescence of the B and the sTRA and CA19-9. So this means the combined effect of this fluorescence B and the fluorescence C, and this sTRA but not CA19-9. This indicates that this should be the result of sTRA minus CA19-9 and this TRA9, that can be seen in this figure. So this clearly indicates that this sTRA occurs independently at different positions or at different locations compared to this CA19-9. So this validates their hypothesis that, or this validates their quotient that sTRA express independently of CA19-9 on the pancreatic tissue. And the quantifications of these results is shown in the panel D, where we can see that in the TMA2, in the case of tumor, the CA19-9 are expressed, CA19-9 sTRA are expressed and are significantly higher than the adjacent tissues, and the CA19-9 and without sTRAs are significantly higher, and the sTRAs but not CA19-9s are also higher compared to the adjacent tissues, and indicating that this CA19-9 and sTRAs seems to be predominantly markers of the pancreatic tissues, not the healthy tissues. And when they did it for TMA5, they see the similar results, that CA19-9 and sTRA are significantly higher, and this CA19-9 only and sTRA only are also CA19-9 only are also significantly higher, but this sTRAs are, in this case TMA5 is not statistically significant compare is not statistically significant compared to the adjacent tissue, but it is higher than the adjacent tissue. And for this TMA6 they do not see any significant results, and for this TMA5 also, and for this TMA5 they shows the results in terms of this nuclear staining, to show that their staining is done properly or evenly. In the panel we can see that the results where CA19-9 and sTRAs are shown, and the CA19-9 only results are shown that CA19-9s are present, and the sTRAs only are also present. And we can see that when the CA19-9s are present where the sTRAs are not present, and for the sTRAs only we can see that sTRAs only are present where CA19-9 are not present, but for the both we can see that sTRAs and CA19 signals can be seen in both the cases. So, and thereafter they check this percentage of tumor cores expresses in either in one or both or neither of the markers. Except for the TMA2 they can see that except for TMA2 section 1 they can see that for the other sections there is an expression of this sTRAs and CA19-9 and sTRA CA19-9, and also this both expressions are also there. For this section 1 the sTRA's expressions could not be found. The reason that they give is that the fluorescence or their method was not optimized in this image acquisition, that is during TMA2 section 1 image acquisitions, whereas for the other case we can see that the sTRAs are also present along with this CA19-9 and the both results were also shown. So this shows that percentage of cores that is only sTRAs, percentage of core that is CA19-9, and some areas are overlap and some of the areas does not have anything. So this completely makes sense for this TMA5 results, where it shows that sTRAs are present but CA19's sTRAs are present. Only sTRAs are present, but these only sTRAs are not present in the adjacent. However, when in the case of this dual, that is CA19 sTRAs, we can see that it's statistically significantly higher compared to the case of the adjacent tissue, indicating that this sTRAs also seems to be a predominantly markers for the pancreatic tissue, and the reason for differences in the different tissue microarrays is because of the different expressions of the pancreatic tissues because of, due to the inherent different mutations that occurs in the patients. I hope you understood how they made a point that sTRAs express independently of the CA19-9 in the pancreatic tissues. If you need to understand more about this results or these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.